Top 1. The coelacanth. A museum curator is sorting through the morning's catch on a South African dock when something halts her entirely. It is December 22nd, 1938, and the scent of fish, salt, and low tide is everywhere. Ordinary until it isn't. There in the pile, 5 feet long and steel blue and iridescent, is a creature with fins that don't move like fins. They move like arms. Marjorie Courtney Latimer, curator of the East London Museum, has never seen anything like it. Neither has anyone alive. According to every fossil record on Earth, the last of its kind died 65 million years ago. She sketched it before it could decompose in the December heat and sent the drawing to ichthyologist J.L.B. Smith at Rhodes University. Smith identified it with trembling hands by his own account as a coelacanth, a species known only from fossils and believed extinct since the Cretaceous-Paleogene boundary. He later wrote that the discovery hit him like a physical blow. It should have. What Courtney Latimer had found on that dock didn't just surprise science. It quietly broke a 65 million year silence. The coelacanth order, coelacanthiforms, has a fossil record stretching back 400 million years. It predates the dinosaurs by 170 million years. It survived the Permian extinction, the Triassic extinction, the Devonian extinction, and then it disappeared completely from every sediment layer, every rock formation, and every dig site on the planet at the Cretaceous boundary. And then it turned up at a fish market. But the gap in the record is only the beginning of the strangeness. When researchers examined the living animal closely, its anatomy revealed something the fossils had only suggested. Its paired lobe fins contain bones homologous to the humerus, radius, and ulna of tetrapod forelimbs. They move in a diagonal sequence, front left, back right, back left, that precisely mirrors the walking gait of a four-limbed land animal. The coelacanth also retains an intracranial joint, a hinge inside its skull, allowing the upper jaw to lift independently of the brain case. No other living vertebrate has this. It was previously known only from early fish fossils. And rather than a fully ossified spine, the coelacanth retains a hollow, oil-filled notochord, the embryonic precursor to the vertebral column, a feature present in the earliest vertebrate fossils and in no other living adult vertebrate on Earth. In 1997, a second living population, Latimeria menadoensis, was discovered off Sulawesi, Indonesia, on the opposite side of the Indian Ocean. This suggested a range far wider than the creature's near-total absence from the fossil record implied. And in 2013, Chris Amamiya and colleagues at the Benaroya Research Institute published an analysis in Nature, finding that the coelacanth genome evolves at the slowest confirmed rate of any vertebrate ever sequenced. Its protein-coding genes have changed less in 400 million years than those of sharks, which are themselves considered evolutionary conservatives. The 65 million year fossil gap even has a name in paleontology. It is called a Lazarus taxon, a species that vanishes from the sediment record and reappears. The coelacanth holds the record for the longest confirmed Lazarus gap of any vertebrate. Paleontologists argue this gap is a preservation artifact, not a true absence. Coelacanths are believed to have retreated to deep-water rocky cave habitats where sedimentation rates are extremely low and fossilization correspondingly rare, possibly near zero. The creature's survival in this view is attributed to the relative stability of the deep Indian Ocean across the events that devastated shallower ecosystems. But 65 million years is not a small gap to attribute to poor preservation. The coelacanth is a large fish, up to 2 meters long and up to 90 kilograms. Large vertebrates leave bones. Bones fossilize. Even deep water species appear in the record through sediment transport in geological uplift. For the entire span from the Paleocene through the Pleistocene, across 65 million years of geological record that has yielded fossils of thousands of other marine species, not one coelacanth scale, tooth or bone has been found. That is not a preservation gap. That is an absence that needs its own explanation. Some researchers in evolutionary biology have asked whether the coelacanth's record-slow genomic mutation rate might itself be the clue, whether this animal possesses mechanisms for genetic stability that actively suppress the mutation rate driving both evolution and eventually extinction. Others have noted that its lobe fins and intracranial joint make it the closest living model of the fish that first walked onto land, and asked whether its survival across 400 million years, essentially unchanged, simply means it found the optimal configuration for its environment so early that nothing has yet improved on it. We may never know which explanation is closer to the truth. What we know is this: the coelacanth survived by hiding in the deep. What comes next survived by doing something far stranger, by stopping at will, being alive at all. Top 2. The tardigrade. In September 2007, European Space Agency researchers loaded several thousand animals into a small container, attached it to the outside of a satellite, and opened it to the vacuum of space. Hard vacuum, cosmic radiation, ultraviolet light 1,000 times the dose lethal to a human. 10 days later, they brought the container back to Earth. Most of the animals were still alive. Some had reproduced in space. The tardigrade was first described in 1773 by German pastor and zoologist Johann August Ephraim Goeze, who called it Kleiner Wasserbär, little water bear, for its lumbering eight-legged gait under a microscope. Italian biologist Lazzaro Spallanzani formally named the phylum Tardigrada the same year. For 2 centuries, they were regarded as biological curiosities. Then researchers began systematically testing their tolerances, and the results progressively dismantled every assumption about the upper boundary of survivable conditions for animal life. The 2007 Photon M3 mission, organized by Ingemar Jönsson and colleagues at Kristianstad University and published in Current Biology in 2008, confirmed what the data had been suggesting for years. These animals can survive conditions that do not exist anywhere on the surface of this planet. And these are not bacteria. They are not spores. Tardigrades are multicellular animals with organs, muscles, a nervous system, and a full reproductive system. When conditions become hostile, they enter a state called cryptobiosis. They expel nearly all body water, reducing it to approximately 1% of normal, retract their limbs, and become a dehydrated form called a ton, in which metabolic activity drops to 0.01% of normal. Biological time effectively stops. In this state, they withstand temperatures ranging from 272 degrees, 1 degree above absolute zero, to positive 150 C. That is a thermal range of 422 degrees that no other multicellular animal approaches. They survive radiation doses up to 570,000 rads of X-ray exposure. The human lethal dose is approximately 500 rads, making tardigrades roughly 1,140 times more radiation-resistant than we are. In 2016, Takekazuo Kunieda and colleagues at the University of Tokyo published genome analysis in Nature Communications, identifying a protein unique to tardigrades called DSSUP, damage suppressor, that physically wraps around DNA and shields it from radiation-induced strand breaks. No equivalent protein has been found in any other animal. That same year, Megumi Sugimoto and colleagues at the National Institute of Polar Research in Japan, publishing in Cryobiology, successfully revived tardigrades from museum specimens that had been stored dry for over 30 years. And the phylum itself appears in the fossil record as far back as the Cambrian period, approximately 530 million years ago, placing tardigrades as survivors of all 5 recognized mass extinction events, including the Permian event that eliminated an estimated 96% of all marine species. Biologists argue that cryptobiosis is an extreme but mechanistically comprehensible adaptation. The DSLP protein, now partially characterized, provides a concrete molecular explanation for radiation resistance. The evolutionary logic is that tardigrades occupy thin-film water habitats, moss, leaf litter, soil, that routinely dry out, imposing survival pressure that over hundreds of millions of years selected for increasingly extreme desiccation tolerance, with other tolerances supposedly emerging as side effects of the same core mechanism. But a protein that physically wraps around chromosomes to block radiation damage is not an accidental side effect of surviving dry conditions in a moss patch. It is a specific, architecturally sophisticated molecular tool serving one function: protecting genetic material from a category of damage that a moss-dwelling animal has no terrestrial reason to encounter. The radiation dosages the DSLP protein is built to resist do not exist on the surface of this Earth. They exist in space. And in 2019, when a lunar lander carrying tardigrades crashed onto the moon, astrobiologists publicly debated whether some of them may have survived the impact and remain viable on the lunar surface today. Some researchers, including astrobiologist David Warmflash, have asked whether tardigrades' capabilities exceed what Earth's evolutionary pressures could plausibly have produced, and whether the DSUP protein demands an explanation that terrestrial natural selection alone cannot supply. Others now treat the tardigrade as the primary model organism for panspermia research. The hypothesis that life or the seeds of life can travel between planetary bodies. If an organism can survive hard vacuum, extreme radiation, and high-velocity impact, the question of where it originally evolved becomes, according to some researchers, genuinely open. From low Earth orbit, we descend into a sandstone canyon in the Blue Mountains of Australia, where a park ranger rappelled into the present and found himself surrounded by the deep past. Top 3. The Wollemi Pine. On September 10, 1994, a park officer named David Noble was rappelling into an unexplored canyon west of Sydney when he reached the floor and looked up. He was standing in a grove of trees he had never seen before. Nobody had. The pollen of this tree appears in rock formations from the Jurassic period. The last time it was recorded alive anywhere on Earth, the continents were still moving into their modern positions. Noble was an officer with the New South Wales National Parks and Wildlife Service, and he had descended into Wollemi Canyon on a recreational bushwalking trip. He collected a foliage sample and brought it to the Royal Botanic Gardens in Sydney. Botanists there identified it as belonging to the family Araucariaceae, but to a genus with no living representative in any botanical collection or field survey on Earth. Pollen from the Wollemi pine genus appears in the fossil record from approximately 200 million years ago. The species was formally described as Wollemia nobilis in 1994 by Ken Hill, Wynne Jones, and Jan Allen, named in part for its discoverer. Noble had found it in one of the most botanically surveyed national parks in the Southern Hemisphere. It had been there the entire time. The wild population at the time of discovery consisted of fewer than 100 mature trees in 2 small groves within a single canyon system, making it among the rarest trees on Earth by population count. Genetic analysis of that population by researchers at the Royal Botanic Gardens, Sydney, found something extraordinary. The entire population is essentially genetically identical. The levels of uniformity are consistent with descent from a single individual or an extremely small founder group, indicating a severe historical bottleneck at some point in the species' recent past. The tree's physical appearance is described by botanists as strikingly prehistoric. Its bark has a distinctive bubbled texture unlike any other living tree. Its branches produce multiple simultaneous growing tips, a pattern called polyaxialism found in no other living conifer. Its leaves are arranged in 4 ranks simultaneously, another feature unique among living species. And the Wollemi pine can reproduce both sexually via cones and clonally, sprouting new shoots from root systems, a mechanism that may allow a single genetic individual to persist indefinitely without successful seed dispersal. The canyon itself maintains a microclimate of high humidity and stable year-round temperatures, sheltered from bushfire by its sheer sandstone walls, forming a refugium that appears to have buffered the species through the climate upheavals of the Pleistocene. The wild location remains classified by the New South Wales government to prevent human disturbance and the introduction of pathogens. Conservation propagation programs now maintain cultivated populations across the world. Botanists describe the Wollemi pine's survival as a refugium effect. The canyon provided a micro-environmentally stable pocket that supposedly insulated the species from the vegetation changes and climate fluctuations that eliminated it everywhere else. The near-zero genetic diversity of the wild population supports the view that it passed through a severe bottleneck and survived through clonal reproduction. Its absence from the fossil record for the past 2 million years is attributed to the rarity of fossilization in the specific sandstone geology of the Blue Mountains. But the genetic uniformity that science describes as bottleneck survival is also a biological alarm signal. In conservation biology, a population with essentially 0 genetic variation has near zero capacity to respond evolutionarily to new pathogens, climate shifts, or novel environmental pressures. The Wollemi pine did not survive 200 million years through robustness. It survived by being hidden. And the question that follows is not only how it survived the past, but whether it can survive us. Some botanists have raised the question of whether the Wollemi pine's clonal reproduction capacity represents a fundamentally different survival logic from the genetic diversity model that dominates conservation biology. If a single genetic individual can persist through vegetative reproduction without theoretical limit, the grove in the canyon may be, in a biological sense, one organism that has been continuously alive for a span of geological time. We cannot determine whether it is a species approaching extinction or a single entity hiding from the Jurassic is not, according to some researchers, entirely settled. The Wollemi pine survived by making itself unreachable. The next creature survived by making itself indispensable. Top 4. The horseshoe crab. Every injectable drug and vaccine manufactured in the United States, every intravenous bag, every spinal injection, every flu shot must pass a contamination test before it enters a human body. That test exists because of an animal that has been walking out of the ocean onto the same beaches for 445 million years. Its blood is blue. Its body plan has not changed since before there were land animals to observe it. And modern pharmaceutical science still cannot replace what it does. The horseshoe crab had long been known as a curiosity, a marine arthropod more closely related to spiders and scorpions than to true crabs. In 1956, Frederick Bang at the Marine Biological Laboratory in Woods Hole, Massachusetts, discovered that horseshoe crab blood clotted almost instantly when exposed to bacterial endotoxins, the specific contaminants most dangerous in injectable medications. This observation led to the development of the limbless amoebocyte lyticate (LAL) test, now the global pharmaceutical standard for contamination detection, mandated by the United States Food and Drug Administration, the European Medicines Agency, and equivalent regulatory bodies worldwide. The horseshoe crab lineage extends to at least the Ordovician period, approximately 445 million years ago. Fossil specimens from the Jurassic period, roughly 150 million years ago, are morphologically indistinguishable from individuals alive today. And in the time between, this animal survived the late Ordovician extinction that took approximately 85% of species, the late Devonian extinction that took approximately 75%, the Permian Triassic event that eliminated approximately 96% of all marine species, the Triassic-Jurassic extinction, and the Cretaceous Paleogene event. All 5, every one. Its blood is copper-based rather than iron-based, hemocyanin rather than hemoglobin, turning blue when oxygenated. Its amoebocyte immune cells produce a clotting agent called coagulagen that responds to bacterial endotoxins in concentrations as low as 1 part per trillion. Approximately 600,000 horseshoe crabs are harvested annually along the United States Atlantic coast for biomedical bleeding, with mortality rates estimated at 10% to 30%, contributing to documented population declines in some regions. A 2019 study published in PLOS One by Owings and colleagues found that bled horseshoe crabs showed altered migratory and spawning behavior even after apparent physical recovery, suggesting sublethal effects extending well beyond the procedure itself. No synthetic replacement for the LAL test has yet received full regulatory approval for routine use, though recombinant factor C is currently under evaluation. And the horseshoe crab, remarkably, has no immune memory, no antibodies, no adaptive immune response of any kind. Yet its innate immune system, unchanged for 445 million years, detects bacterial contamination at concentrations that defeat modern analytical chemistry. Evolutionary biologists describe the horseshoe crab's persistence as a case of ecological generalism and physiological robustness. It tolerates wide ranges of temperature and salinity, and its benthic seafloor lifestyle supposedly insulated it from surface extinction events. The body plan stability is attributed to a fitness plateau. The organism is highly optimized for its niche, and the niche has remained stable across geological time. But fitness plateau describes the outcome, not the mechanism. To maintain genetic stasis for 445 million years across 5 catastrophic extinction events requires either an extraordinarily stable environment across all of them, which the geological record does not support, or a biological mechanism that actively resists heritable change. The horseshoe crab's genome is large and complex, not minimized like a highly adapted specialist. It has retained its architecture across a time span in which every other complex animal on Earth transformed beyond recognition. That is not optimization. That is something else, and we do not yet have a name for it. Some researchers in evolutionary genetics have proposed that certain lineages may possess what has been called evolutionary stasis mechanisms, genomic or developmental regulatory architectures that buffer against accumulating viable mutations, not by preventing mutation, which is inevitable, but by making most mutations developmentally lethal, preserving the phenotype. If such mechanisms exist, some researchers believe the horseshoe crab's genome may be one of the clearest places to look for them. Others have noted the more uncomfortable version of the same question. Why does modern synthetic chemistry, with complete knowledge of the coagulagen mechanism, still lack an approved replacement after 60 years of trying? From the tidal flats of the American Atlantic, we go deep. Top 5. The goblin shark past the light line, past the pressure threshold of recreational diving, into the dark where a shark with a jaw that exits its face has been waiting since the Cretaceous. The goblin shark lives in complete darkness, 1,000 meters below the surface in water, cold enough to kill a human in minutes. It has skin so translucent you can see the blood vessels beneath it, lending it a permanent pink-gray pallor that looks less like coloration and more like exposure. And when it detects prey, its jaw, the entire lower face, extends outside its skull independently and explosively and snaps shut on whatever is in range. This is not a creature that appears to have been refined by time. It appears to have been designed by something that had never seen a constraint. The goblin shark, Mitsukurina ostoni, was first brought to Western science in 1898 by Japanese naturalist Alan Austin, who passed a specimen to marine biologist David Starr Jordan. Jordan named it for Kakichi Mitsukuri, the Japanese zoologist who had drawn his attention to it. Fossil teeth indistinguishable from those of living goblin sharks have been found in Cretaceous deposits approximately 125 million years old. Since 1898, fewer than 50 complete or near-complete specimens have been collected in all of recorded scientific history. The species' behavior in the wild, its reproductive biology, and its population size remain, to an extraordinary degree, unknown. The goblin shark belongs to the family Mitsukurinidae, of which it is the sole living member. Its closest extinct relatives, the genus Scapanorhynchus, are known from the Cretaceous and are near identical dentally to living specimens. Its jaw mechanism, termed slingshot or snap-jaw feeding, operates through highly elastic tendons and ligaments that store and release energy explosively, projecting the entire jaw assembly forward by up to 9.4% of total body length in under 0.3 of a second. This was measured in the only detailed kinematic study of a living specimen ever conducted, carried out by Nakaya and colleagues, and published in PLS 1 in 2016. That slingshot jaw mechanism is found in no other living shark species. It is found in no other living vertebrate. Its evolutionary origin within a single shark lineage is not well understood. The goblin shark's snout contains an exceptionally dense array of ampullae of Lorenzini, the electroreceptive organs present in all sharks, suggesting sensory tuning specifically for detecting the electrical fields produced by prey in conditions of total darkness and zero visibility. Its skin is described as extremely delicate relative to other sharks, its translucent quality consistent with a lineage that has never been subjected to surface ultraviolet radiation. The International Union for Conservation of Nature lists it as least concern based on the absence of evidence of decline, a classification that researchers have noted is functionally equivalent to an admission that the species is too poorly monitored to assess. And in January 2007, approximately 100 goblin sharks were incidentally captured by prawn trawlers off southeastern Australia at depths of 40 to 60 meters, shallower than any previously recorded specimen, an event that has not been explained or replicated. Marine biologists classify the goblin shark as a bathypelagic specialist, a deepwater ambush predator whose extreme morphology reflects adaptation to hunting in a low-energy, lightless environment. The protrusible jaw is explained as a mechanism for prey capture without full-body acceleration, conserving energy where food is sparse. Persistence since the Cretaceous is attributed to the comparative stability of deep benthic environments during and after the KPG impact. But the KPG event did not spare the deep ocean. Ocean acidification, thermal disruption, and the collapse of the phytoplankton base of the marine food web following the impact affected deep-water food chains globally, and numerous deep-water species went extinct at the KPG boundary. Why this specific lineage persisted when others in similar environments did not remains genuinely unexplained. And given that fewer than 50 specimens have been collected across all of recorded scientific history, the honest position is that we do not have enough data to explain the goblin shark's survival, its current population, or even confidently whether it is endangered. Some marine biologists have raised the possibility that the goblin shark's near absence from the scientific record reflects not rarity, but distribution in zones that systematically avoid standard sampling depths. Its 2007 Australian shallow water appearance, the first confirmed shallow record in the species' history, suggests depth flexibility not previously documented. If the animal's actual range is substantially broader than 50 specimens imply, some researchers ask whether what we call a rare survivor might simply be a creature we have never had the equipment or the opportunity to properly find. The goblin shark survived by going where no one follows. The next animal survived by outlasting every relative it ever had. Top 6. The tuatara. As the sole survivor of an entire order that once spanned multiple continents, watching the world from a rocky island at the edge of the Pacific with 3 eyes, the tuatara. On a small island at the northern tip of New Zealand's South Island, in the cold dark of the Cook Strait, a reptile sits motionless and watches the sky. It watches with 3 eyes. Its order flourished alongside the earliest dinosaurs, more than 60 genera, more than 100 species spread across every continent except Antarctica, and every one of them is now a fossil. It has been the last one for approximately 60 million years. It does not appear to have noticed. The tuatara, Sphenodon punctatus, was described by John Edward Gray of the British Museum in 1842. Its superficial resemblance to lizards led to its classification among them for decades until 1867, when German biologist Albert Gunther recognized it as the sole surviving member of the order Rhynchocephalia, an order abundant globally during the Triassic and Jurassic with over 60 genera and more than 100 species, which had declined to a single species confined to New Zealand's offshore islands. By the late 20th century, the mainland New Zealand population had been eliminated by introduced mammals. A conservation program established in 1989 by the New Zealand Department of Conservation, including predator eradication and captive breeding, stabilized the surviving island populations. The fossil record of Rhynchocephalia is global. Specimens have been found on every continent except Antarctica. The last non-New Zealand Rhynchocephalian fossils date to approximately 60 million years ago in South America, meaning this order persisted on multiple landmasses for 180 million years before disappearing everywhere except one island chain in the southern Pacific. The tuatara's parietal eye, the third eye, sits on top of the skull beneath a scale that gradually becomes opaque in the first months of life. The structure contains a lens, a primitive retina with photoreceptors, and a connection to the pineal gland. It does not form images in adult animals, but its precise function in circadian regulation has not been fully confirmed. The tuatara is the only living reptile with a single row of upper teeth fitting between 2 rows of lower teeth, a configuration shared with certain fossil rhynchocephalians and found in no other living vertebrate. As the teeth wear down over a lifetime, individuals eventually chew with bare bone and then shift to gum chewing entirely. Females reproduce only once every 2 to 5 years. Incubation takes 11 to 16 months, the longest of any reptile. Individuals do not reach sexual maturity until 10 to 20 years of age. One captive individual at the Southland Museum in New Zealand named Henry successfully reproduced at an estimated age of 111 years. Their preferred active body temperature of 16 to 20 degrees is far lower than all other reptiles, and they remain active in temperatures down to 7 degrees Celsius, tolerating cold better than any other ectotherm. In 2020, a genome sequencing study published in Nature by Neil Gemmell and colleagues found the Tuatara genome to be approximately 5 gigabases in size, 60% larger than the human genome, containing gene families associated with immunity, smell, and vision that show evidence of rapid evolution within an otherwise profoundly conserved body plan. Genetic analysis confirmed the Tuatara lineage diverged from the common ancestor of all living lizards and snakes approximately 250 million years ago, before the Triassic-Jurassic extinction and before the breakup of the supercontinent Pangaea. Biogeographers attribute the Tuatara's survival to New Zealand's geographic isolation. The landmass separated from Gondwana approximately 80 million years ago without land bridges, preventing the arrival of the placental mammals that outcompeted rhynchocephalians globally following the Cape extinction. In this view, the Tuatara survived not through competitive advantage, but because its island ecosystem was shielded from the pressures that eliminated everything else. But New Zealand's isolation does not explain why rhynchocephalians were already declining globally throughout the Cretaceous, 60 million years before the final extinction event. Something else was reducing them long before the KPG boundary, and the Tuatara's genome, 60% larger than a human's, carrying gene families showing active molecular evolution within an ancient body plan, suggests an organism carrying biological complexity that has not been fully characterized. Some evolutionary biologists have asked whether the Tuatara's persistence involves the same genomic architecture hypothesized for the coelacanth and the horseshoe crab. Conserved developmental genes that preserve body plan alongside active evolution in specific functional gene families affecting survival. Whether that mechanism is real and whether it is identifiable remains, according to those researchers, an open question. From a rocky island in the southern Pacific, we traveled to the tropical forest floors of 3 continents simultaneously. Top 7, the velvet worm, to find an animal that has occupied all of them with the same body since the Cambrian explosion 500 million years ago. The velvet worm. Look closely at the leaf litter of a tropical forest floor in Central America, Sub-Saharan Africa, or Southeast Asia, and you might find it. A creature that looks like a caterpillar, upholstered in velvet, moving on up to 43 pairs of unjointed legs that leave a soft ripple trail in wet soil. When it detects prey, it turns and fires 2 jets of adhesive slime from glands flanking its mouth. Slime that sets on contact with air and immobilizes a cricket or woodlouse in seconds. Then it bites through the slime and feeds. It has been doing this with this exact body since the Cambrian explosion, the single most transformative event in the history of animal life 500 million years ago. Velvet worms, phylum Onychophora, were first described to Western science in 1826 by surgeon and naturalist Lansdowne Gilding from a specimen collected in Jamaica. For decades, they were misclassified, placed at various times with annelid worms, arthropods, and mollusks before their true phylogenetic position was clarified through comparison with Cambrian fossils. The discovery of hallucigenia and related Cambrian organisms in the Burgess Shale of British Columbia and their interpretation by Simon Conway Morris in 1977 revealed that the velvet worm body plan was already present in Cambrian seas 530 million years ago. The living velvet worm is a near-direct descendant of those Cambrian ancestors, having migrated from marine to terrestrial environments without fundamentally altering its architecture. The velvet worm occupies the phylogenetic position that bridges the 2 most species-rich animal phyla on Earth, Arthropoda and Annelida, and has held that position for 530 million years without resolving into either. Its unjointed hydrostatic legs, termed lobopods, contain fluid-filled body cavities that extend under pressure rather than through rigid joint articulation. Comparative developmental biologists, including Graham Budd at Uppsala University, have used the velvet worm leg as a model for reconstructing the evolutionary transition from soft lobopod to rigid arthropod limb. The slime ejection system fires a protein-based adhesive at distances up to 30 centimeters with controllable directional accuracy used for both prey, capture, and predator defense, and breaks down rapidly after use, leaving no environmental residue. Velvet worms also have a circulatory system structurally intermediate between the closed circulatory system of vertebrates and the open hemicoel of arthropods. A partially partitioned hemicoel, considered a transitional form between the two systems. They breathe through trachea, branching tubes opening through pores in the skin, a respiratory system convergently evolved with that of insects, supporting their classification as a sister group to arthropods. A 2021 phylogenomic study published in Current Biology by Oliveira and colleagues confirmed that the divergence of Onychophora from the arthropod stem lineage predates all currently known arthropod fossils, placing the split at approximately 550 million years ago or earlier. And despite spanning multiple isolated continental populations, suggesting ancient Gondwanan distribution across what became separate landmasses, all known velvet worm species share the same fundamental body plan across the entire transition from marine to terrestrial environments, multiple continental separations, and 530 million years of biotic upheaval. Evolutionary biologists describe the velvet worm as a successful generalist in stable microhabitat niches. Moist tropical and subtropical leaf litter and rotting wood that have persisted relatively unchanged since the Carboniferous. The body plan's persistence is explained as a fitness plateau in a stable environment. The soft-bodied fluid locomotion design works in wet leaf litter and has never been displaced by a superior competitor in that specific niche. But a moist leaf litter specialist should have been seriously disrupted by the Permian extinction, which caused global aridification severe enough to devastate terrestrial ecosystems alongside marine ones. That the velvet worm survived on multiple continents simultaneously with the same body plan intact across all of them suggests either that the Permian catastrophe left isolated forest floor refugia intact across the entire former Gondwana landmass, or that the velvet worm's physiology possesses a resilience to environmental disruption that has not been systematically examined. Some researchers have proposed that the velvet worm body plan may occupy what is termed a developmental attractor state, a configuration so internally consistent that mutations disrupting it are systematically selected against, not because the environment punishes change, but because the developmental system itself resists it. Whether that constitutes a real biological mechanism or a description in search of one remains, according to those researchers, an open and important question. From a creature that has held its position between two kingdoms for 500 million years, we turn to the one animal that watched its closest relatives go completely and permanently extinct and kept swimming unchanged in the same ocean where they vanished. Top 8. The Nautilus. 66 million years ago, an asteroid struck the Yucatan Peninsula and changed the chemistry of the oceans for thousands of years. In the extinction that followed, ammonites, 10,000 species strong, 300 million years of fossil record, one of the most successful groups in marine history, went completely and permanently extinct. Their shelled cousins, the nautiluses, swimming in the same oceans, eating similar food, carrying a nearly identical body plan, survived without interruption. Science has not produced a fully satisfying explanation for the difference. Nautiluses have been known since ancient times, their shells collected across the Mediterranean and Asia for millennia. The living animal was formally described by Linnaeus in 1758. By the 19th century, the nautilus's role as the last surviving nautiloid cephalopod and its relationship to the extinct ammonites was well established. The critical scientific mystery solidified as the KPG boundary was documented in increasingly precise stratigraphic detail across the 20th century. The complete extinction of ammonites and the complete survival of nautiloids became one of the most examined puzzles in marine paleontology, studied most extensively by Neil Landman at the American Museum of Natural History. Ammonoidea is documented in the fossil record from approximately 400 million years ago and diversified into approximately 10,000 described species across the Devonian, Carboniferous, Permian, Triassic, and Cretaceous. The final ammonite fossil occurs precisely at the KPG boundary 66 million years ago, with no confirmed post-boundary survivors anywhere in the record. Nautiloidea is documented from approximately 500 million years ago. Far more diverse in the Paleozoic, the group declined to a handful of coiled genera by the Mesozoic and currently consists of 4 living species of Nautilus and 1 species of Alunautilus. The primary hypothesis for differential survival was developed by Landman and reviewed in Paleobiology in 2007. It centers on egg size. Ammonites are believed to have produced small planktonic larvae floating in surface water, while nautiluses produced large, yolk-rich eggs laid on the seafloor, hatching as miniature adults capable of immediate benthic life. The KPG surface productivity collapse would have devastated planktonic larvae while leaving seafloor-dwelling juveniles comparatively intact. Nautiluses also take 5 to 10 years to reach sexual maturity, live 15 to 20 years, and produce approximately 10 to 20 large eggs per year. An unusually conservative life history for a cephalopod, most of which reproduce once and die. The nautilus shell implodes at approximately 800 meters depth, confining the animal to a depth band of 150 to 600 meters in reef slope environments. Genetic analysis has described the nautilus genome as showing comparatively low rates of molecular evolution in housekeeping genes, consistent with the living fossil genomic pattern documented in the coelacanth and the horseshoe crab. And then there is the population data. A 2016 study by Peter Ward and colleagues at the University of Washington, published in PLOS 1, documented population declines of up to 80% in Nautilus pompilius across parts of its Indo-Pacific range over 15 years, driven by shell trade and bycatch. An animal that survived 5 mass extinctions is now facing a human-driven crisis within decades. The Landman egg size hypothesis is the most widely accepted explanation for why nautiluses survived while ammonites did not. Deep habitat combined with benthic egg strategy provided supposedly just enough buffer to pull the lineage through the surface collapse. But the egg size hypothesis is inferred, not observed. No ammonite eggs have ever been found. The hypothesis rests on analogy with modern cephalopods and inference from shell structure. And even if ammonite larvae were planktonic, the KPG event did not uniformly destroy the surface ocean. Some marine groups with planktonic larvae survived. The question of why specifically this lineage crossed the boundary, rather than why planktonic larvae were generally disadvantaged, remains exactly as open as the fossil record makes it. Some paleontologists, including David Jacobs at UCLA, have asked whether the nautilus's conservative life history constitutes a general survival strategy for mass extinction events, rather than a specific KPG adaptation. Whether nautiluses survived not because they were positioned correctly in 66 million BC, but because their life history profile systematically buffers against the kinds of ecological disruption that mass extinctions produce. Whether that constitutes a documentable principle of extinction biology or simply describes what happened in one well-studied case is, according to those researchers, an open and important question. An animal that has survived 5 mass extinctions is struggling to survive us. The next creature on this list has survived every extinction event in the geological record for 520 million years, without changing a single visible feature of its body. Scientists have a name for what it does. They do not fully agree on why it works. Top 9. Lingula. Take a living lingula from a tidal flat in Japan and place it beside a fossil from the Cambrian explosion, the event 520 million years ago, when nearly every modern animal body plan appeared simultaneously in the record. Measure the shell dimensions. Examine the lophophore structure. Compare the anchoring pedicle. They are not similar. They are the same animal. In 520 million years, through 5 mass extinctions, through the breakup of supercontinents, through the rise and extinction of the dinosaurs, through the entire evolutionary history of every vertebrate that has ever lived, Lingula changed nothing at all. Lingula was first formally described by Jean-Baptiste Lamarck in 1801 from Indo-Pacific specimens. Its Cambrian fossil record was established throughout the 19th century. In 1952, paleontologist Norman Newell at Columbia University published an influential review designating Lingula as the type example of morphological stasis, the most extreme documented case of evolutionary non-change in the Metazoan fossil record. Its survival through all 5 recognized mass extinction events has made it the standard reference point in discussions of extinction selectivity for over 70 years. Cambrian specimens from the Chengjiang biota of Yunnan, China, approximately 520 million years old, were studied by Jianhan and colleagues at Northwest University in Xi'an and published in Science in 2007. They show lop preservation and shell morphology indistinguishable from living Lingula under light microscopy. The animal belongs to the phylum Brachiopoda, superficially resembling bivalve mollusks but fundamentally distinct. Its 2 shell valves are dorsal and ventral rather than lateral, and its feeding organ is a lopophore, a crown of ciliated tentacles that creates water currents to filter food particles. It uses a phosphatic shell composed of calcium phosphate rather than the calcium carbonate used by almost all modern shelled marine animals, a chemical composition that was common in the Cambrian but is now extremely rare among living invertebrates. Lingula lives in a U-shaped burrow in soft intertidal sediment, anchored by a flexible muscular pedicle that extends and retracts to maintain position. This lifestyle requires no hard substrate, no reef, and no specific temperature range, a habitat generalism that may contribute to its across-extinction persistence. The Permian-Triassic extinction event devastated the Brachiopoda as a whole, reducing the group from approximately 400 Permian genera to approximately 20 surviving genera crossing the boundary. Yet Lingula crossed that boundary with no documented morphological change recorded in continuous stratigraphic sections in South China by Chen and Benton in their 2012 review published in Nature Geoscience. And in 2020, a genome sequencing study by the Beijing Genomics Institute and collaborating institutions published in Nature Communications found that the Lingula genome shows extreme conservation of developmental toolkit genes, the Hox genes and patterning genes controlling body plan architecture, with significantly lower substitution rates in these genes than in other invertebrate phyla, suggesting active maintenance of developmental architecture rather than passive coincidence. Paleontologists attribute Lingula's stasis to stabilizing selection in a stable habitat. The intertidal mudflat niche has existed in various forms since the Cambrian, and the current body plan is supposedly so well adapted to it that any deviation is selected against. The phosphatic shell, pedicle anchoring, and lopophore feeding system form an integrated functional unit that works and has always worked in the conditions it occupies. Extinction events that disrupted surface and reef ecosystems left intertidal mudflat niches comparatively intact, allowing generalist burrowers to persist through events that devastated specialized fauna. But the 2020 genome study's finding of reduced substitution rates in developmental toolkit genes suggests something more specific than a coincidence of habitat stability. To say that Lingula is unchanged because change is selected against is, in a sense, to explain the survival by the survival. The genomic evidence suggests an active biological constraint on the rate of heritable change in the genes most directly responsible for body plan architecture. Some researchers have proposed that Lingula's phosphatic shell chemistry may also have conferred a passive advantage during extinction events driven by ocean acidification, specifically that phosphate-based shells may be more stable than carbonate shells under the acidification conditions produced by both the Permian and KP boundary events. In this reading, Lingula's survival is not despite being primitive. It is, according to some researchers, because of it. Lingula has held the same body for longer than any other animal we can document. The last creature on this list has been in the ocean for longer than flowering plants have existed on land, and we have never seen one alive long enough to study it properly. Top 10. The frilled shark. In the 16th and 17th centuries, sailors returned from deep ocean voyages with accounts of a creature they had glimpsed at the surface, eel-like, long as a ship's boat, with a frilled neck and a body that moved through dark water like something from before the world had rules. They were dismissed as liars or as men whose minds had buckled under months at sea. In 1884, a Japanese marine biologist pulled one from the waters off Numazu and sent it to Harvard. The sailors had not been lying. The sea serpent was real. It was just very, very old. The frilled shark, Chlamydococcus anguineus, was formally described by Samuel Garman at Harvard in 1884 from specimens provided by Japanese zoologist Kakichi Mitsukuri. Its morphology was so divergent from all other known sharks that Garman placed it in its own family, a classification that has stood largely unchanged since. Fossil teeth indistinguishable from those of living frilled sharks have been found in Cretaceous deposits approximately 80 million years old. Since 1884, fewer than 50 complete or near-complete specimens have been collected in all of recorded scientific history. A second species, Chlamydocelicus africana, was described from South African waters only in 2009. The frilled shark belongs to the order Hexantiiformes, the most ancient surviving shark order, with Hexantiiform fossils documented from the early Jurassic, approximately 175 million years ago. Proposed affinities with Carboniferous sharks would extend the lineage past 300 million years, though this remains contested. Its 6 gill slits, all other modern sharks have 5, are covered by a continuous frilled membrane on the first gill arch. 6 gill slits is a primitive character retained from early shark evolution and subsequently lost in every other modern shark order. Its 300 teeth are each tricuspid, 3-pointed, arranged not to cut or grip, but to trap soft-bodied prey, squid and fish, in a basket from which escape is effectively impossible once the jaw closes. Gestation in the frilled shark was estimated at approximately 3.5 years in a 2012 study by Tanaka and colleagues at Tokai University, published in PLOS One, using embryo-sized data from specimens caught by Japanese fishermen. 3.5 years. The elephant gestates for 22 months. The frilled shark's gestation period is the longest confirmed vertebrate gestation on record by a significant margin. In January 2007, a living frilled shark was observed and filmed in Awashima Marine Park in Japan after being found swimming near the surface in a harbor, behavior completely anomalous for a deep-water species. The animal died within hours. The incident has not been explained. A 2016 study published in PLOS One by researchers at the Portuguese Institute for the Ocean and Atmosphere documented frilled shark bycatch in Portuguese Atlantic waters at depths of 700 to 1,000 meters, noting that most specimens were juveniles, suggesting possible shallow water nursery areas not previously recorded anywhere in the literature. The International Union for Conservation of Nature lists the frilled shark as least concern. Researchers have noted that this classification is based not on population data, but on the absence of evidence of decline, an absence that reflects the near-total absence of population monitoring of any kind. Marine biologists classify the frilled shark as a deep-water ambush predator whose eel-like body allows efficient movement through rocky, complex deep-sea terrain. The protrusible jaw and basket tooth architecture are explained as adaptations for capturing fast, soft-bodied prey without full-body acceleration, conserving energy in a habitat where food is sparse. Survival across the KPG boundary is attributed to the comparative stability of deep benthic environments during and after the impact event. But the frilled shark's gestation period is not just long by deep-sea standards. At 3.5 years, it is the longest confirmed gestation of any vertebrate ever recorded. In a reproductive cycle that long, combined with a population size we cannot quantify and a bycatch rate we cannot monitor, means that recovery from any significant population disruption would take decades at minimum. The oldest shark lineage on Earth, an animal that supposedly survived every catastrophe the planet has produced, is being pulled up in fishing nets at a rate that cannot be assessed because we have never collected enough individuals to establish a baseline. Some researchers have asked whether the frilled shark's morphological conservatism may conceal molecular evolution in immunity, sensory, and reproductive genes, the same pattern found in the coelacanth, the tuatara, and lingola, in which body plan genes are conserved while specific functional gene families continue to evolve. If the frilled shark genome follows this pattern, it would, according to some researchers, reinforce the hypothesis that living fossil survival reflects a specific and identifiable genomic architecture shared across ancient lineages. The problem is that the frilled shark's genome has not yet been fully sequenced and published. One of the oldest shark lineages on Earth, a potential window into Cretaceous marine biology, remains genomically uncharacterized because specimens are too rarely collected and too fragile to provide samples of adequate quality. We have known about this animal for over 140 years. We still know almost nothing about it. 10 creatures, 10 survival records that span from the Cambrian to the present. 10 cases where the standard explanations, stable habitat, geographic isolation, ecological generalism, deep water refuge, account for a pathway to survival without fully accounting for the precision of the outcome. The coelacanth vanished from 65 million years of fossil record and turned up in a fish market. The tardigrade carries a protein that shields DNA from radiation levels that do not exist on Earth's surface. The Wollemi pine survived in a single canyon, while one of the world's most botanically surveyed parks surrounded it for decades. The horseshoe crab's immune mechanism, unchanged for 445 million years, is still the only approved test for pharmaceutical contamination. The goblin shark has a jaw that exits its face and fewer than 50 confirmed specimens in over a century of scientific awareness. The tuatara carries a genome 60% larger than a human's and a third eye with a functioning retina on top of its skull. The velvet worm has held a phylogenetic position between two animal kingdoms for 530 million years without resolving into either. The nautilus watched 10,000 ammonite species go completely extinct and kept swimming. Lingula has a genome in which the genes controlling body architecture show substitution rates that statistical analysis has difficulty distinguishing from zero. The frilled shark has been gestating its young for longer than any other vertebrate on record and has never been observed alive long enough to study. The mainstream explanation for each of these is reasonable. It is probably partly correct. Deep water stability is real. Island isolation is real. Niche generalism is real. These are legitimate factors in survival. What they do not fully explain is the pattern, the specific repeated appearance across deeply unrelated lineages of near-zero change in body plan genes, combined with apparent active evolution in functional genes, combined with survival across events that reset the rest of life on Earth. That pattern has a name. It is called a living fossil, but the name describes the observation, not the mechanism. The mechanism, if it exists as a coherent biological principle, has not been identified. Some researchers believe it will be found in the genome, in conserved developmental toolkit genes, in regulatory architectures that buffer against heritable change, in proteins like TSUP that exceed what terrestrial selection pressures should have produced. Others believe each case is simply what it appears to be, a lucky animal in a stable place, surviving by coincidence across deep time. We may never know which explanation is closer to the truth. What we do know is that these 10 animals are the best available evidence that the question deserves to be asked. Most people pass them in a tidal flat or museum display and see nothing unusual. The textbook explanation fills the space before curiosity can form. But the gap between what the fossil record says and what these animals require is not closed by the explanations we currently have. That gap is where the interesting questions live. On December 22, 1938, a woman stood on a South African dock and looked at something that should not exist. 65 million years of silence. One fish. The entire fossil record quietly, completely wrong. The 10 creatures in this video have been here longer than our language, longer than our science, longer in some cases than the ocean floor beneath them. They did not survive to be explained. They survived because something in them, something we have named but not yet understood, refuses to stop. Some questions are not answered, they are inherited. If these ancient survivors have changed the way you look at the history of life on Earth, please subscribe to the channel and like this video. We are uncovering the biological secrets that time tried to bury. Help us grow the future of discovery. Our next deep time treasure will be revealed soon.